Thank you very much. I also would like to thank Bram, Bert, Helge for inviting me and for generating this wonderful meeting in this wonderful city, I have to say. And I will proceed with another question for patients. So
hello, my name is Peter, 6 years old.
And my name is Johannes. I am Peter's father. And we have 2 questions for you. The first one is, could you provide a timeline on your rollout phase of the clinical trials? And the second one,
how can I help you?
how can we as patients contribute to drive your projects into a successful direction? Thanks a lot.
Bye-bye.
So, very, very easy questions. It's easy to answer, I guess. I will try. So we move a little bit. I will say something that is already touched today and also yesterday. And I wanted to start with the expectation for a cure, of course, that is everyone here in the audience and at home is expecting for. I want to touch the natural history study that we heard yesterday also about, the patient registry that was introduced also by Gustavo today, and what is the path to the clinical trials. So first of all, I have to start with what Madeleine depicted very well. So we started some years ago thinking about a problem only in the muscle, so that was called myosin deficient muscular dystrophy. So nobody was interested in other tissues, but now we know that it's more complex. So not only the muscle is involved, but also the nerve is involved, the brain is involved, gastrointestinal system is involved, those patients have a problem in growth, they have a problem in bones, possibly they have a problem in heart, so it's more complex. And when the more is complex the disease, also the more is complex to arrive to a therapy. And or, on the other side, we have to know where to deserve the therapy in the proper tissues. And the good news, also, Madeleine introduced you, we have many animal models, and this is not always for all the genetic disease. In this case, we have at least 3, and now there is a new one, she presented animal models. They cover almost many aspects of the disease and the different complexities, so this is a good ground to start from. Then, of course, we will have organoids, cells, and what else, but we need to have complex models in order to test our therapies. And everything starts from, of course, the absence of this molecule that we now we know everything about that. And the problem, again, is a multi-organ disease. This is a structural protein, but also a functional protein, so it's more easy, for example, to replace an enzyme. It's more difficult to replace a structural protein in general in the body. And it is also a very large gene and a large protein, and it makes many difficulties. Of course, we may replace the gene and the protein, but since it's very big, we still have a problem with tools to do it. So probably it will be solved in the future, but at the moment, this is still something which is not really possible, at least at the clinical level. We can use compensation system, and it was shown how it could be managed in previous talk, and we can modulate other pathways. So the protein has many functions, the system is disrupted, so as Madeleine says, we may modulate, for example, apoptosis or the control of cell cycle or how the metabolism works. So those are also introducing possibly a way to delay the progression of the disease. So we are cycling towards the top of the hill, but still we have many things to do. And those things really relies to the fact that even if we have a drug to test, we need to be prepared to do it. And this is not so easy. So when we start to think about a drug, we think that it's very easy. You have a drug, then you have the patients, you give the patient the drugs, and that's it. You will see and you resolve everything. But this is not probably so easy as we expected. So we have many things to obtain, first of all. And one of those things is to measure what we are going to evaluate. So you see, we need a natural history study to measure what we want to see is going to change if we apply a drug. And this is extremely important because it's the only way in which we understand if the drug is working or is not working. And if we spend years trying with a drug is not working, then it's loss of time, of course. What we can measure. So we can measure many things. We can measure the strength of the muscle. It's easy. So if we know how it moves towards in the typical life of a person. So you see, if it's in this way, it's quite easy. If I know that after a certain time point he will be here or will be here, we can insert the drug and to see if it changes. But it probably is not always the same for all the patients. We have to know, for example, at which age we have a change of a specific function. Of course, ambulation is much easier to test, but also how to raise the hands could be another thing we can test, why we use the hands. So we may measure many things. In this case, we have to measure the progression of respiratory problems. We can measure the development of contractors, so the limitation of the range of motion. But we have to really understand if there is a variability between patients. Otherwise, all our measurements will fail in a certain way. So the problem is if we have high variability. And in fact, we already learned that it is. So we know there are patients with very severe form, and we know we are patients who have a milder form, but now we understood that there are patients in between. So it is heterogeneous, the number of patients that we see. They have a different mutations, so we are trying to understand if there is any correlation between the clinical phenotype and the type of mutation. You have seen yesterday that we have some hints that may suggest it is, and we have a different progression in the speed. So I may deteriorate a lot in a period of time and then being stable for another period. So if I want to test the drug, probably I have to choose the right period to test this drug. So here I'm introducing the fact that there is a heterogeneity in the patients. We have to stratify the patients. We have to define definitely a good subgroup of the patients in order to serve them the right drug. And this is possible only if we know how the disease moves, so if we know very well natural history. So let's see what we learn from other diseases. In this case, I'm talking about Duchenne muscular dystrophy. So I think you all know Duchenne muscular dystrophy. We think the patients are more or less very similar one each other. They have the progression of the disease, and the progression is more or less this one. So we expect that when they start to have first symptoms, about 3-4 years old, and then they decline a little bit and progressively until they lose, for example, ambulation. This is what we think is the disease, but this is what is the reality. So all these lines are different patients. Here they are measuring a 6-minute walking test, so how many meters they can walk in 6 minutes. And so you expect the medium is this one, so it's similar what we expected to see. But you see, all these patients behave in a different way. And you can subgroup these patients. And you see there are patients with a fast decline, patients with a moderate decline, patients that are stable, patients that even increase for a certain period of time the capacity, the motricity. And you may expect that it's very different if you treat patients in this group or in this group. So you may completely not understand what's going on with your drug. So if you treat patients which are ameliorating, you think your drug is always working. If you treat patients that deteriorate, you may think that your drug is not working. So you have really to choose which kind of patient to treat. And for example, if you have time, so if you are, for example, you have more than 1 year, 2-3 years of time to decide if your drug is working, you may expect to treat the patient along this line. But if you have to treat them in 1 year, you have to treat those patients exactly in this window. So because you know that in this window, they decline with a certain shape, otherwise you will lose probably the possibility to understand anything. And this is extremely important. And this is what the regulators ask to clinicians in general when they go there, or to companies when they go there and they want to have the possibility to test a new drug. So they want to have, of course, a validated outcome measures and when to apply those validated outcome measures. So they want to have exactly in which window those clinicians or companies want to measure that outcome measures is going to deteriorate, and if the drug is working, they are going to stop this kind of deterioration. So they also want to know if it's meaningful what they are going to measure, because of course, as I said, you may measure the decline in 6-minute walking test, and you may have a possibility to see that you change the shape. But now we know that maybe you change the shape because they deteriorate 5 meters less in 1 year. So this could be significant from the statistical point of view, but maybe have no clinical significance. So it doesn't change anything in the life of a patient. Instead, if you can change, for example, 50 meters in this test, definitely you are going to change the life of the patient. So the quality of life is getting better. So this is what we want to know. So we want to know which kind of outcome measure and how much we need to change this outcome measure in that period to have a clinical significance to your patients. This could be a 6-minute walking test, but could be, as you see, measurement of the upper limbs or the other scales that have been introduced, for example, for LAMA2 disease. So we need very sensitive scales. We need to know how they change, how it changes the measurement, and if it is meaningful. Okay, so the other thing is that we also require a size of patients we want to analyze. And this is a problem, especially for very rare diseases such as this one. You see, we have listened yesterday about natural history, mainly focusing on 20, 30, 40 patients, and this is a very small size, and this could be a problem because what we measure in a limited number of patients could not reach significance. So maybe we are testing something that is good, but we cannot really demonstrate with a number that is going to be a good drug. So what we need to have a natural history probably in a larger number of patients in order to have a measurement that give us a quantitative number which is more appropriate, and in order to be able to correspond what we see in our patient when we give the drugs, if they are changing or not with respect to that. And this is also important because when you're talking about rare diseases such as this one, if you, for example, if you want to apply a drug and you can recruit only 20, 30 patients, it's very difficult to have a double-blind study. So it's very difficult to serve to some patient the drug and to serve to other patient the placebo, because the number is so few that either the drug is, as I said yesterday, is a miracle. Otherwise, it's difficult to measure the difference. So we need natural history studies which are very precise in order to know exactly in that group of patients how they move. And so probably we may apply for open studies in which all the patients are treated and we can compare what's going on in those patients as respect to the history. And this was already done, for example, again, in Duchenne muscular dystrophy. You see here that this was the line of patients not taking a drug, but obtained for a natural history study. Those were specific patients amenable for skipping exon 51 and with a specific number of meters at the 6-meter walking test. And they were compared with those patients taking the drug. So those are the patients not taking a drug, these are taking a drug. So you see they could measure a difference, which is significant. In this case was patients taking a drug. This was the natural history, so they could compare the results. And based on this comparison, the FDA gave the approval for a drug. And so in the United States, for example, they could use this drug. So that's why it's important to have a very well-designed natural history studies, precise, in order to be potentially compared to what's going on in a clinical trial. The other thing is to have the possibility to have a biomarker or surrogate biomarker, and this could be anything. So we have heard about blood, we heard about MRI, it could be neurophysiological evaluation, so it doesn't matter. We need something that corresponds to a progression of the disease and can tell us, maybe in a shorter period of time, if we are doing something good on those patients or not. Because, for example, maybe measuring deterioration of strength will take too long for a clinical trial. Instead, measurement of a specific biomarker, so a protein in the blood, if it's going to change something, it will be observed much earlier. And this, for example, is what's going on for a disease which is called amyotrophic lateral sclerosis. This is a severe disease of the central nervous system, again with degeneration and defects in motricity. What they found is in the blood there is a rise of this protein, which is called neurofilament, is a cytoskeleton protein in the neurons, and patients with ALS have an increased amount of neurofilaments in the blood. And the more they have disease, the more the level is high. But the more interesting thing is that even if they have a mild, a moderate, or a very severe disease progression, the evaluation of this protein is stable in time. So it means that if I have a good drug that can apply, and I apply the drug at a certain point of the life of my patients, I can see if I can move this line down. If it moves down, it means that I'm reducing this biomarker and probably I'm doing something good for the patients. And so that's why probably we need a biomarker, even in this case, so in this disorder, to be more reliable in what we are going with the drug to our patients. So the other problem with natural history studies, as I said, is that many natural history studies have been started or are going to start in different countries. We have the Dutch study, which started a few years ago. We heard about the Swiss study. We heard about the French study, which started last year. The Spain started in 22. In Italy started this year, practically. They're all going to serve us some information, but as I said before, the number of patients into those studies probably will be too small, so the size will be too small to have then very significant results. So whatever we see will be good, whatever we see will give us a hint in how to proceed, but probably we did something which is more a common ground in which to evaluate our patients. So probably we need supranational studies, and those supranational studies will give the possibility to include many patients. We have seen that there are hundreds of patients around the world. Of course, it's not easy to do, but this will be the only way probably to obtain significant results to be measured and then to be used in clinical trials. The other thing is that it's very difficult for us to share data from our studies. So the data I obtain in Italy is difficult. I can share those data with those in Spain, on the UK, for many reasons. The first one is for regulatory points of view. So all the ethical committee will stop this kind of sharing unless we have a common ethical approval and we have a common frame in which we obtain the data. So I think one of the things we have to do probably is try to build up a natural history study supranationally, so in which we can, of course, include many patients. And to do that, probably we have to introduce again the problem of the patient registry. This is still a crucial event because I think I put the registry together with a natural history study. I think they run in parallel. We need both of them to be successful in our activity. And it's the only way in which we can really identify how the disease progresses, how complicated it is, what is the safety we have to look for our patients, so how we can build all the information necessary for our study. And this is also a way to make the disease, as it is, more attractive for companies. As someone told this morning, the first thing they ask when they come to a community is how many patients do you have with these kinds of aspects we can treat, because of course they invest money in a drug. They invest money not because of philanthropy, but because of course they want to obtain other money. And so it could be a win-win situation because they want to have money and we want to have a therapy. So it's good to collaborate with them, but we have to serve them the best as we can. So we have to be able to give them all the information. We have X number of patients at this age or at that age, X number of patients that they can ambulate or cannot ambulate, and so far. So those numbers have peripheral neuropathies, those numbers has heart problem, and so far. So we need to have this information, and to have this information, the good recipient is the registry. And the registry is also a way to improve the standard of care to the patients and to make them to be part of the discussion when we build up a study or when we build up anything that is devoted to you. Again, here there could be many kinds of registry. We may have a single national registry or instead a single multinational registry. There are pros and cons in all of this. To be honest, I think that single national registry, they are important in order to know where there are the patients, but they are not so much important in order to collect the data. So we need to have a registry in which we can collect the data from patients all over the world. So we can discern whenever it's possible to all the patients the possibility to participate to a clinical trial, even if they live in a country where the trial probably is not going to be started, maybe started in the country aside, so they can be alerted and come to the country in which the study is ongoing. Of course, there are complexity in doing this kind of registry because there are many regulations, different languages, different ownership of the data, but I think this is something we have to approach. And there are many kinds of supranational registry. We heard about QCMD registry. There is one for Europe, which is the European registry for the EARN, so the network of excellence for rare disease in Europe. Again, there are pros and cons. It's very well done. The one from QCMD is coming from the patients, but probably the data inserted is not so precise that can be used in clinical trials. On the other side, for example, the EARN registry could be more precise because all the data will be inserted by the physicians, but for example, only some of the institutes are allowed to enter in this network, so not all the patients will be represented. So we have probably to force to find a registry which works in both directions. And so what is our path to clinical trials? And here I will be quite fast. So we need again, a good natural history study and a registry. We need to decide which kind of patient we want to treat, because it may be different for different drugs. As I said yesterday, if you want to apply gene therapy, probably you need very young patients. You cannot apply gene therapy at the moment in adult patients, but if we want to do exon skipping, maybe we can apply also to adult patients, or if we have a small molecule that interferes with apoptosis, we may apply to all the patients. So depending on the patients and the range, we have to build up our design. We need a good preclinical safety and toxicological data coming from preclinical studies and then also in humans. You need GMP production, and which is not a trivial situation. GMP production is very expensive and could be done only in certain situations. And so this is something we have to think about, why maybe some of the trials are not so fast to arrive to patients. Then we have to be approved by the regulatory system, otherwise you cannot start a study, and then you need funding, which is another point which is extremely important, because those studies are very expensive. I don't want to spend too much time then in which kind of outcome measure we have seen already today and yesterday. We have many possibilities in the ongoing natural history study. They are testing all those outcome measures. Probably we will have to match the data to see if one of these is, or which one of these is significant. And also we cannot apply all this kind of outcome measure, because otherwise we have to keep the patients days in the hospital, and this is not possible. So concluding, those were the questions of Peter, and those probably are the answers. So we are not far away, but still we have some street to do. And I try to serve you all the reason why we have some things to do still. And what you can do as a patient, definitely you have to force your clinicians to make you part of a natural history study and to belong to registries. Otherwise, you are not known, and if we don't know you and we don't know how you behave, we lost some items. And I think I stayed in time.
Thank you. Thank you very much, Stefano. This was great. And we have time maybe for one question for this talk, or we can continue discussion at the round table. I cannot see any hands up, and there are no questions from the chat. There was a question for the previous talk, but we can continue later. Thank you so much, Stefano. Thank you.